This industrial case study explores the topic of lithium-ion batteries, laboratory measurements, within the framework of the European project titled Personalized Education in Measurement Science and Technology Empowered by Machine Learning. Two types of analysis can be performed on lithium-ion batteries in a laboratory: non-invasive and invasive. Non-invasive methods evaluate the performance by means of electrical measurements of voltage and current. These include discharge voltage curve and its differential analysis, pulse power test, and electrochemical impedance spectroscopy. Invasive methods can be further adopted to analyze the material conditions, but they often require disassembling the cell. They include half-cell voltage curve and differential analysis, SEM, EDS, and XRD. The most performed analysis is the discharge curves characterization. The cell is first charged at constant current up to the maximum voltage, and then fully charged at constant voltage until the residual current becomes lower than a threshold. This starting point is considered the fully charged condition. Discharge curves are measured at multiple current rates and different temperatures. As a general rule, the cell voltage reduces as the current increases or the temperature decreases, because the polarization voltage increases. In fact, at lower temperatures, the internal resistance increases as the chemical reactions become unfavorable. Discharge curves allow the calculation of the cell capacity, which is measured in ampere hours. Starting from the cell discharge voltage characteristics, it is possible to perform differential analysis to highlight the phase change transitions. This analysis offers insights into material degradation. The pulse power test is adopted to measure the equivalent internal resistance of a cell. A current pulse is applied, and the voltage response is measured. The resistance is defined as the variation of voltage divided by the variation of current at a given period, often 1 second or 10 seconds. The resistance tends to increase at lower temperatures as well as with aging, mainly due to the growth of the films at the surface of the electrodes. As the cell degrades, the capacity reduces and the resistance increases. The relationship between capacity reduction and resistance increase is not the same in all aging conditions. For instance, research results obtained after applying fast charge cycles at multiple temperatures have shown that at low temperatures, capacity reduction is the key degradation mode due to lithium plating, while resistance does not increase much. On the other hand, at higher temperatures, capacity reduction is limited, but the resistance increases more. In fact, at higher temperatures, the key degradation mode is the growth of films at the surface of the electrodes, causing an increase in the series resistance. The resistive and diffusive behavior inside a cell can be characterized by an electrical circuit composed of resistances and capacitances. The parameters of the equivalent circuit can be estimated by charging and discharging the cell with a dynamic current stimulus, such as a square wave. Electrochemical impedance spectroscopy is often adopted to gather further insights about the resistive and diffusive behavior caused by each electrode. The cell is stimulated with an alternating current signal at multiple frequencies, and the resulting complex impedance is plotted on a Nyquist plot. The data is fitted to an equivalent circuit, allowing the calculation of the equivalent circuit parameters. The electrochemical impedance spectroscopy characteristics depend on 3 key factors: temperature, state of charge, and aging. In fact, lower temperatures imply higher resistance due to the uneasiness of chemical reactions because of lower thermal energy. In general, the resistance reduces at higher states of charge. Moreover, aging plays an important role in resistance. As the cell degradation proceeds, the resistance increases. As a consequence, the internal resistance of a cell dramatically increases in the condition when an aged cell is operated at low temperature and low state of charge. On the other hand, the best performance is obtained with a fresh cell at ambient temperature and a high state of charge. Electrochemical impedance spectroscopy can also be employed in dynamic conditions, such as while charging the cell at a relatively low current rate. This analysis allows us to observe the evolution of the internal impedance with the state of charge. To evaluate the influence of degradation on each electrode separately, half cells can be assembled with metallic lithium as negative electrode. Material analysis can also be performed by means of SEM, EDS, and XRD methods. SEM and EDS are often adopted to observe the presence of cracks on electrode particles and material contamination. XRD is employed to analyze the crystalline structure. Thank you for your attention. This video was produced through the collaboration of SS Cyril and Methodius University of Skopje, the University of Niš and Politecnico di Milano, within the framework of the European project Personalized Education in Measurement Science and Technology Empowered by Machine Learning. More information about the project can be found on the project website and LinkedIn page.